Hey friends, welcome back to my channel. Hope you all are fine and safe. So our today's topic is plant tissue culture. Now plant tissue culture is the aseptic method of growing cells and organs such as meristems, leaves, roots, etc. either in solid or liquid medium under controlled condition. So it is an in vitro cultivation of all plant parts, whether it is a single cell, a tissue or organ, but under aseptic conditions, under controlled environment, where we can control the temperature and other required necessary condition for the proper growth and development of a plant. Now, who is the father of plant tissue culture? So, Haberland is the father of plant tissue culture. The two below given images in this slide, the first is of a glass bottle and the second is of a laminar airflow. This glass bottle, in this only we will inoculate the explant which will be further growing in this with the further stages of that explant will takes place in this glass bottle, and not only in a single glass bottle. This I have shown you for an example that this glass bottle is used in this plant tissue culture operation, and as the plant will grow, we have to subculture, means we have to change this glass bottle, and in this the media is there. Okay. Now laminar airflow. This laminar airflow is the place where we have to do the plant tissue culture operations aseptically. Now this are the steps involved in the plant tissue culture. So as now we know what is plant tissue culture, we should also know what are the steps that are involved in this tissue culture operation. So first is the selection of plant. What we have to do is, like, the plant tissue culture steps are same for all the things which we will do for all the plants. Somewhat method difference, 5% method difference is there, but for study purpose what we have to write in exam, these are the steps. Okay, now the images which you can see here under the headings of selection of plant, isolation of explant, these images I have put for your understanding. Okay, I have added for your understanding. These are the images of different plants. Okay, so first is the selection of plant of this field is of banana, as you can see. So how we will select a plant? Okay, so what we have to see to select a plant is, when we will go to a field and when we will see that there are hundreds of bananas of plant, more than hundreds of bananas plant, then how can we select a mother plant from which we will have to take the explant for our further plant tissue culture operation? So we will see the morphological physiological features. We have to measure the height, we have to see the color, we have to find the healthy plant, and this is the selection of plant. After this, the isolation of explant. Okay, as you can see here, the isolation of explant, a picture of banana plant is I have added here. So the below portion, this below portion, okay, this below portion we have to cut, and when we will cut this, what we will get is, we will get the below portion of this tree trunk along with the mud and the roots. Okay, then we have to do the sterilization of this explants. Okay, from one banana tree we'll get one single explant. Yes, then sterilization of explant can be done with different methods. The first is the dry heat, the second is the wet heat or autoclaving, then with ultrafiltration, ultraviolet sterilization, flame sterilization, surface sterilization, whipping with 70% alcohol. I think most of the students already have practiced in laboratories with many other plants, whether they are doing any other experiments, this whipping with 70% alcohol. I think everybody should, every student have done this. Okay, now after sterilization, then we will do sterilization. Okay, as I have taken bananas example in these two steps, I'm going to tell you what I have done in my plant tissue culture training, the sterilization process. Okay, so chemical sterilization was opted when I was having training of plant tissue culture in banana. What they used to do, okay, what I have done is, when the explant came, okay, when the people used to get, when they went and bought the explant, have to be washed thoroughly with the this with normal tap water. At many tubs are there. In one tub there will be normal tap water, okay, the next plant will be put in that tap water. We have to clean that, and after that we have to add in that fungicide, bactericide, okay, this bactericide and fungicide, and after it again we have to wash it with the tap water. And after this cleaning, the cutting process will be there, okay, we have to get the smallest size what we want accordingly. The smallest size will get, and after that again we will dip it in a solution containing water, bactericide, fungicide, okay, and then we have to take it to the, before taking it to the lab again, then we have to wash it with normal tap water, and then we have to keep it in the normal jar which we have seen in the previous slide, okay, and then we have to take it in the laboratory under aseptic condition. There we have to again we have to do some chemical sterilization process, and then we can use it in that laminar airflow for cutting and inoculation in the glass jars. Now this is the normal process, okay, which I have done in chemical sterilization. Different plants have different institutes, different private firms have different procedures. Now fourth is inoculation of explant. Now what is inoculation of explant? In this, the explant is placed in autoclaved glass bottles. What glass bottle which we have seen in the that glass bottle we cannot use directly, we have to first autoclave it. After its autoclaving, we can inoculate that explant in that bottle which contains media. Please name that media name, if any of the student knows about it, what is the name of the media which we use in the plant tissue culture, in the comment section below, then I would be happy that students know this, okay. I think at this point of the time you should know that what is that media which we used in plant tissue culture. Now in laminar airflow, after cutting of the explant, the proper size which we want, we will inoculate it in the bottle, okay, glass bottles. Now incubation. Now as you can see in the image, in the growth rooms, okay, growth rooms are there, in which proper light, proper temperature control light setup is there, and there we have to put like this stands. Stands are already built there, and there we have to put our bottles, okay, for some days, so that suppose if we have inoculate the explant, so it is a first stage, and after some time we have to do the second stage. So between the first stage and the second stage, between the second stage and the third stage, and the so on, we have to keep these bottles in the growth room, so that they will have enough time, enough control temperature to grow in a proper condition, to grow properly and have their proper development. Now as you can see this image incubation, this is of some other plant, this is not of first stage of banana. I have just kept it for your information to show you that how the bottles are used to keep bottle in the growth room, okay. Now coming forward to this, coming back to this fourth point, inoculation of explant. As you can see here, this, as you this bottle, okay, so what, as I told you earlier that when we isolate an explant, okay, from a banana tree or any of the plant tree, then we have to cut it in the smallest size which we want it to be. So this is the smallest size of the explant, we have cutted it and we have put it in the media, okay, and after that we can put it in the growth room, that is incubation. Now sixth point is initiation of callus or differentiated organs, for example shoots. Now when you will write in your examination, okay, there you will write the steps, like you will make the flow chart, that steps involved in plant tissue culture. So first is selection of plant, isolation of explant will be second, then sterilization of explants, inoculation of explants, incubation. Sixth point which you used to always find in every books, in every PDF, will only be initiation of callus, okay. As plant tissue culture involves culturing of all plant parts, which can include a single cell, organ, or a single tissue, or anything like culturing of roots, stems, leaves, meristem. So I have added here for your understanding, oblique I have done for callus, that it is differentiated organs, for example shoots, and I have put it first is of first is of the differentiated organs coming like after first stage, after we have inoculated the explant, some growth have been seen, and that growth is that coming of the differentiated organs like shoots are coming. And the second image is of the callus, initiation of callus. If we have done plant tissue culture with a single cell, okay, then we can see that second stage, which is callus. And if we have done with any explant of a plant, then we can see that first image, that is the growing of shoots. Okay, now what is callus? So callus is the unorganized mass of a cell. Now seventh step is multiplication or subculturing. Okay, so what is multiplication or what is subculturing? As you can remember that previously in the first slide I have explained you about subculturing when I was explaining you about the glass bottle, that we have to change the media, means we have to change the bottles, the explant which we have put it in the first bottle. After some time we have to again put that explant when it will have some growth, then we have to put again it in a proper freshly prepared media, that is in the another glass bottle. Okay, so this is subculturing, so that it will get enough space for its further growth and development until it will reach to the last stage. Every plant has its own stage. Banana has 8 stages, bamboo has 15 stages for its complete development until it gets ready for the field establishment. Now multiplication of subculturing, what it involves it is, after some days when explant shows visible growth, then it is transferred to a freshly prepared media to obtain large number of plantlets. Okay, now 8th point is regeneration. So what is regeneration? Further, an entire plantlet can be induced to grow on culture media by manipulating the phytohormone balance correctly. Understood? Phytohormones means plant hormones. If you want a video for phytohormones, then you can write in the comment section below. 9th point is hardening. And students, I'm remembering you again, if you have any doubts regarding this content or want to ask any question regarding this topic, you can ask me in the comment section below. Now 9th point is hardening. So what it is? See, hardening can be of two types. For example, if I'll take example of banana plants, it can be primary, secondary. So for primary hardening, what we have to do is, after the completion of all the stages in banana's growth, what we'll do is, we have to separate that banana plantlets from the media, from the bottle, and then we have to clean it, and then again we have to plant it in a pot tray. Pot tray means that trays are there in which we have to, like you can see that ice cream, not ice cream, you can see that ice trays in your fridges, like that pot trays are there in which we can put the soil mixtures, and then we can make small holes so that the plantlets can fix in that, and we will what potting we do now, like that we will put the plantlets there. After some days, when plants grow and they get ready for the secondary hardening, then we can put it for secondary hardening. What we will do is, we again then put that plants from that pro trays to the black polythene bags, poly bags, or in paper bags, and then we can keep it in the shade house, so that now for some days the plant will get the natural environment and will get ready for the field establishment. Now, what are the common steps of hardening which you can write in your exams? Are this 5? So, what it is? The in vitro cultured rooted plants are first subjected to acclimatization before transferring to the fields. Means we try to make the plants comfortable with the natural environment, and then after it bears all the natural things, then we can send it for field establishment. Second is the plantlet is taken out from the rooting medium, is washed thoroughly to remove entire agar from the surface of plantlet, as agar may attract microbes to grow and destroy the plantlets. Now, what is this agar? Okay, tell me in comment section, from where do we get this agar? Okay, from where do we get this agar? So agar is a solidifying agent, the media in which we use to inoculate our plantlets. That media contains agar which used to solidify the media. Now third is the plantlet is now kept in a low minimal salt medium for 24 to 48 hours and transferred to a pot that contains autoclaved sterilized mixture of clay soil, coarse sand and leaf molds in 1:1:1 ratio proportion. Fourth is the pot containing plantlet is covered generally with the transparent polythene cover having holes for aeration to maintain the humidity. The plantlets are maintained for about 15 to 30 days in this condition. See, now this is the point hardening, in which 5 points are there. You can write this in your examination. The previous which I have explained you, that primary hardening, secondary hardening, this is that which I have explained you. This is an example of banana, that what I have did. Okay. Now 10th point is transfer of plantlets to greenhouse or open field. After doing the hardening proper, we can then transfer that plantlets to greenhouse or open house. So thank you, this is it. Now you can if you have any doubt, then you can ask in the comment section below. Thank you, have a nice day.